Okay, so I'm now going to I'm going to switch gears and I'm going to talk about organellar DNA. So the DNA in our nuclei is not the only DNA that we have. We also have DNA in our cytoplasm. This is often called cytoplasmic DNA, and it's found in our mitochondria, and if we were plants, it would also be found in our chloroplasts. So a couple things about mitochondria and chloroplasts. They're both in eukaryotic cells. They are both they both consist of sort of a double membrane structure going on here. They're both fairly small, and they both have their own DNA, and they also both have their own ribosomes. All eukaryotic cells contain mitochondria. Some eukaryotic cells additionally contain chloroplasts. Oops, sorry about that. Both mitochondria and chloroplasts can make their own ribosomal RNA and transfer RNA, rRNA and tRNA. They both have those genes, but there are about 900 proteins that are found in mitochondria, and most of those are actually encoded by the nuclear genome. And so you can see over evolutionary time that genes that are necessary for mitochondrial function have been lost from the mitochondrial genome and actually made their way onto the nuclear genome. And so the mitochondria have kind of downsized their genomes, and most of what they need are made by the host cell. So the evolutionary origins of mitochondria and chloroplasts are thought to come from endosymbiotic bacteria, and there's a few lines of evidence that point to that. First, eubacteria have a number of physical characteristics with mitochondria that are both physical and genetic characteristics. Second, there are a number of protist unicellular eukaryotes today that actually do have endosymbiotic bacteria, so it's not crazy to think that that was originally the source for mitochondria, since we do know that this happens. And so in theory, we would take some sort of way back when anaerobic eukaryotic cell, it would phagocytose some sort of bacterium, they're calling it a protobacterium. And oh, I forgot to mention that both mitochondria and chloroplasts, they generate energy. They make ATP. Chloroplasts harvest energy from sunlight. Mitochondria use the electron transport chain. Okay, so we start out with an anaerobic cell that takes up an aerobic protobacterium that then starts making energy for it. And eventually, so here is where it branches. Then we can, in one group, we also get cyanobacterium that gets engulfed and incorporated into the host cell, and this represents then becomes our modern day chloroplast, whereas this guy becomes our modern day mitochondrion. So another line of evidence for the endosymbiont theory is that the mitochondrial and chloroplast ribosomes are actually structurally similar to bacterial ribosomes. And so there's similarities between the physical characteristics, the genetic characteristics, as well as their ribosomal characteristics. And finally, there are antibiotics that we can use to experimentally inhibit protein synthesis in eubacteria, but they don't affect eukaryotes. So when we apply these to eukaryotic cells, they inhibit mitochondrial and chloroplast protein synthesis. And so you take a drug that targets bacteria, and it works on mitochondria and chloroplasts. Both mitochondria and chloroplasts are uniparentally inherited. That means that they are inherited from only a single parent, and usually it's mom. So we get all of our mitochondria from our moms, and so we have a maternal line of descent of our mitochondria. Plants, it's the same thing, where chloroplasts are inherited from the maternal parent. So mitochondria are present in sperm, and they enter the egg during fertilization, but then they're later targeted for destruction. And there are some exceptions to this rule. There are bivalves, which include oysters, clams, and mussels that we like to eat, have biparental inheritance. So in those organisms, mitochondria inherited from mom and dad. In gymnosperms, which we know as conifers, and a few angiosperm that are flowering plants, they have actual paternal organelle inheritance rather than maternal. So there is some variation along this theme. So as a result of the uniparental inheritance that generally occurs, there is no recombination for mitochondrial DNA. And so mitochondria are generally clonal in nature. So variation arises in mitochondria due to mutation. And when this happens, so what you're seeing here is a cell, and all these little dots are different mitochondria. So a cell can contain many different mitochondria. These mitochondria have a replication cycle that's independent of the cell's replication cycle. And so you can have a single mitochondrion giving rise to multiple mitochondria within the cytoplasm of a cell. If we have a mutation in one of these mitochondria and it divides and occupies part of the cell, you end up with what's called heteroplasmy. And cells, these are cells that contain mitochondria or chloroplasts with different mutations and thus sort of genetically different organelles. And this can result in what's called replicative segregation. So here we have a heteroplasmic cell that we start out with. And during cell division, the cell is super careful about equal allocation of the nuclear genetic material to both the daughter cells, but it's less careful about equal allocation of cytoplasmic genetic material to the daughter cells. And so different daughter cells can end up, if you have a heteroplasmic cell, different daughter cells can end up with different proportions of here the red mitochondrial type versus the blue mitochondrial type. Over time, over multiple cell divisions, and just through random sampling, really, of mitochondria to among the daughter cells, sort of random segregation, you can end up with multiple cell divisions down the line, generations of cell divisions down the line. You can end up with these descendant cells that all have different proportions of the different mitochondrial types, and in some cases they've only got one mitochondrial type or the other mitochondrial type. And this process of random allocation, such that you start out with a heteroplasmic cell at some ancestral position, and the descendants end up with different mitochondrial genotypes, that is replicative segregation. So this can be seen in certain types of diseases that are caused by mitochondrial defects. And one example is myoclonic epilepsy and ragged red fiber syndrome, which is abbreviated as MRF. And individuals with MRF have varying degrees of severity of their symptoms depending on the proportion of affected mitochondria that they actually have. So there's been some attention, I don't know if you've noticed or not, but there's been some legal debate in the news over the idea of the three-parent baby, where there are women sometimes who have mitochondrial deficiencies and don't want to pass those on to their children. And so there's an IVF technique that's, that basically allows the mother to donate her nuclear genetic material, but not her mitochondria. And so I'll leave it to you guys to pause the video and look this over more carefully if you want to figure out exactly how they do this. But what ends up with is that you have three parents. You have the mom's nuclear genome, you have the dad's nuclear genome, and then you have a third woman's mitochondrial genome within the egg. So, and there have been a number of court hearings regarding legality and such of this technique. Okay. So what the mitochondrial genome looks like is it's small. Mitochondrial DNA is circular, much like most bacterial DNA, and there in some species mitochondrial DNA is linear, and there's also some species of bacteria in which mitochondrial DNA is linear also. And so again, variation along this theme. There's always exceptions to every rule. All right, each mitochondrion carries, so so a cell has lots of mitochondria, potentially. Each mitochondrion can have a number of, like, multiple copies of its genome. You can think of it similarly to how bacteria can have multiple copies of plasmids. Mitochondrial DNA does not have histones, but the DNA is organized with other kinds of proteins, just like bacteria have. And the size of mitochondrial DNA varies from species to species. Along this, here's the table just to give you a sense for the level of variation you can have. Most species have about 40 to 50 mitochondrial DNA genes. Plant mitochondrial DNA tends to be large, so as you can see down here, and variable in size. And in angiosperms especially, mitochondrial DNA contains these long repeats in these large non-coding regions, which is kind of unusual. And it can cross over with itself to, here's the mitochondrial. So here's all of our mitochondrial DNA genes. Here's our D loop. This is where our origin of replication is located. And then a whole bunch of other stuff going on in there. Plant mitochondrial DNA gets kind of crazy because it has these repetitive regions that allow crossing over to occur, and sometimes you can get multiple chromosomes that kind of pop out over evolutionary time, which is kind of crazy. Okay, and because mitochondrial DNA doesn't recombine in the same way that the Y chromosome can be used to trace male ancestry, the mitochondrial genome can be used to trace female ancestry. And so a couple things about mitochondrial DNA that make them really amenable to genetic studies is that each cell contains really only a single copy of the nuclear genome, but many, many, many copies of the mitochondrial genome. And so mitochondrial DNA is often easier to isolate and amplify from samples that, DNA is fairly stable as a molecule, but even it degrades over time. But mitochondrial DNA can often be recovered from samples where you can't get nuclear DNA. So that's one advantage to it. And in animals, mitochondrial DNA evolves relatively quickly. And it makes it a good marker for resolving evolutionary relationships of fairly closely related species or populations. And then the maternal inheritance of mitochondrial DNA, again, it lacks recombination. And so then you can trace lineages of, for example, here humans, to kind of understand human evolution. And so mitochondrial DNA was used as a method for supporting the out of Africa hypothesis of human evolution.